Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 8th December 2020. And as all of us know that we are into the revision mode, we are revising the entire syllabus, and as part of that we are revising chapter number 14, Respiration in Plants. And in our previous session we discussed an all-important concept called glycolysis. As I said, lysis means breaking. Glyco refers to glucose. So glucose is getting broken down as part of respiratory reaction. In this respiratory reaction, namely glucose, we have been understanding that a molecule of glucose having 6 carbon atoms will be broken down to glucose 6-phosphate with energy ATP being spent and forms ADP, adenosine diphosphate plus inorganic phosphate, and glucose 6-phosphate then becomes fructose 6-phosphate. Again ATP molecule is spent and ADP and inorganic phosphate are formed. That means in the first 2 steps of glycolysis, 2 ATP molecules are spent. So keep remembering this, 2 ATPs are spent. And then further, the fructose 1,6-biphosphate having 6 carbons split into 2 molecules, 3 carbon plus 3 carbon, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And these 2 chemical compounds are reversible. The glyceraldehyde 3-phosphate having 3 carbon will form 1,3-biphosphoglyceric acid. This is very extremely important step, as there is formation of very powerful chemical highly energetic molecule called NADH2. That is, NAD becomes NADH plus H plus. And this is a profit in this reaction. And 1,3-biphosphoglyceric acid becomes 3-phosphoglyceric acid. Again, it's an important step because ADP plus inorganic phosphate becomes ATP. This is the first ATP molecule that is produced here. And 3-phosphoglyceric acid becomes 2-phosphoglycerate or glyceric acid. And 2-phosphoglycerate becomes phosphoenol pyruvate PEP, and phosphoenol pyruvate becomes pyruvic acid, the end product of glycolysis. And phosphoenol pyruvate PEP forming 3 carbon pyruvic acid produces another ATP molecule. So ADP plus inorganic phosphate becomes ATP. That means overall, if you see in glycolysis, 2 ATPs are used. Where are those 2 ATPs are used? As I said, glucose becoming glucose 6-phosphate, 1 ATP is used. Fructose 6-phosphate becoming fructose 1,6-biphosphate. So 2 ATPs are used. And 4 ATPs are produced. Where are those 4 ATPs produced? Step number 1, 1,3-biphosphoglyceric acid. When it is produced, let me show you here. 1,3-biphosphoglyceric. Yeah, you can see here, when 1,3-biphosphoglyceric acid producing 3-phosphoglycerate, you can see the formation of ATP molecules. Since 2 molecules are involved, 2 ATPs are produced. And then another one is the step phosphoenol pyruvate, PEP, becoming pyruvic acid. That is 2 ATP. So you got here plus 2 and this also plus 2. 2 plus 2, 4 ATPs are produced, and here minus 2 ATPs. Okay, so you are having net gain of plus 2 ATPs. Plus, what is important is there is formation of NADH. 2 molecules of NADH are produced. Where is it? This one. 2 molecules of NADH. This is highly powerful molecules. 2 molecules of NADH, and these two molecules of NADH is equivalent to 6. That means 1 NADH is equal to 3 ATP molecules. So 2 into 3, 6 ATP molecules. So overall, at the end of the glycolysis, net gain is 6 plus 2, 8 ATP molecules. This is the profit for glycolysis. So there is a net gain of 8 ATP molecules during the process of glycolysis. So the scheme of glycolysis is given by 3 scientists. It's called EMP pathway, very famous as EMP pathway. Emden, Meyerhoff, Parnas, EMP. So only it has become famous as EMP pathway. Glycolysis is partial oxidation of glucose or similar hexose sugar into 2 molecules of pyruvic acid through a series of enzyme mediated reaction releasing some ATPs and NADH. It occurs in the cytoplasm. In plants, glucose is derived from sucrose or from storage carbohydrates. Sucrose is converted into glucose and fructose by enzyme invertase. Glycolysis starts with the phosphorylation of glucose in the presence of an enzyme hexokinase to form glucose 6-phosphate. 1 molecule of ATP is used in this process. In next step, glucose 6-phosphate is converted to fructose 6-phosphate and that is catalyzed by phosphohexoisomerase. And fructose 6-phosphate uses another molecule of ATP to form fructose 1,6-biphosphate in the presence of an enzyme phosphofructokinase. That means in the glycolysis, every step that is formed will be mediated by an enzyme. So you need to remember the enzyme names. So pyruvic acid is taking part in these 3 steps: lactic acid formation, alcoholic fermentation, and of course aerobic respiration. So in glycolysis, 2 molecules of ATP are consumed during double phosphorylation of glucose to fructose 1,6-biphosphate. 2 molecules of NADH are formed at the time of oxidation of glyceraldehyde 3-phosphate to 1,3,1,3. Yes, NADH, it is 1,3-biphosphoglyceric acid. Each NADH is equivalent to 3 ATP, so the net gain of glycolysis is 8 ATP as explained to you just now. Pyruvic acid is the key product of glycolysis. What is the end product of glycolysis? It is always pyruvic acid. And the further breakdown of pyruvic acid depends upon the need of the cell. In animal cells like muscles, during exercise, when oxygen is insufficient for aerobic respiration, pyruvic acid is reduced to lactic acid by an enzyme lactate dehydrogenase due to reduction of NADH2. That's how we get what is called muscle fatigue. Now, how is that happens? This is how. That is, the glucose molecule and glyceraldehyde 3-phosphate is formed. Glyceraldehyde 3-phosphate becomes 3-phosphoglyceric acid, and here NAD plus is becoming NADH, a rich energy molecule is formed, and 3-phosphoglyceric acid is forming phosphoenol pyruvate. And this phosphoenol pyruvic acid, forms when oxygen is not available in rich state, or when oxygen is absent in the muscles, it will form lactic acid. So pyruvic acid getting converted to lactic acid, now a rich NADH molecule is converted back into NAD. That means we are losing energy. So when the muscle loses energy and lactic acid formation gets accumulated, muscle fatigue comes. That's how athletes, when they are running, suddenly they collapse and they complain of muscle catch. That muscle catch is because of this lactic acid. This is very common in sports people, where they do not warm up, and without warming up, if they directly take part in the game, the lactic acid formation will be there in the muscles, and muscles become fatigued, muscles become tight, and once the muscles become tight, mobility, movements will be restricted. Sometimes there may be muscle tear also. And this pyruvic acid also forms in the presence of fungi, ethyl alcohol, ethanol, and here again NADH, high energy compound, is being spent. NADH is becoming NAD+. So this process is called fermentation. So in fermentation by yeast, pyruvic acid is converted to ethanol and CO2. The enzyme involved is pyruvic acid decarboxylase and alcohol dehydrogenase catalyze this reaction. In both lactic acid fermentation and alcohol fermentation, very less amount of energy is released. Yeasts poison themselves to death if concentration of alcohol reaches above 13%. So final products of glycolysis, pyruvate is transported from cytoplasm into mitochondria. This is important. Pyruvate is transported from cytoplasm to mitochondria for further breakdown. Oxidation of pyruvate to acetyl-CoA is done to produce CO2 and NADH. The reaction catalyzed by pyruvic dehydrogenase requires participation of several coenzymes including NAD+. So pyruvic acid, coenzyme A, NAD+, in the presence of magnesium and pyruvate dehydrogenase enzyme becomes acetyl-CoA plus CO2 and NADH is formed, and all important. So the acetyl-CoA enters in a cyclic pathway called TCA cycle or Krebs cycle for further breaking the pyruvic acid. So acetyl-CoA is very, very important, and that's how the Krebs cycle starts. We continue this aerobic respiration further in our next session.